Sure, sure. Please
Yeah.
go ahead.
Okay, I'm just starting it here. Okay. Yes. So, so this is the latest review that has been published. It's published on, I think, April 20th, 7th, April 17th. Yeah, April 7, right? So, and it is a beautiful, I mean, yeah, we can write even better review for this, but we need to understand what this paper covered. So this paper is about the oligonucleotides and COVID-19. So it says like the advantages of the oligonucleotides are three, which is capacity of rational drug design, then swift developmental cycles, then pursuing undruggable targets, right? Then it explains like the COVID-19 is caused by SARS-CoV-2, the severe acute respiratory syndrome, coronavirus 2, which is closely related to, but it is not identical to coronaviruses, which caused SARS and MERS, Middle Eastern Respiratory Syndrome. So there are like, it says like there are 12 compounds under investigation for the treatment of COVID-19, but there is still no definitive treatment available as of now. So the classes of the compounds are like anti-HIV drugs, then anti-malarial drugs as hydroxychloroquine. Then there are some experimental antiviral compounds, and also people are using the antibody-rich plasma from the infected people. So what is the role of oligos? We can develop, so there are two strategies in which we can use the oligonucleotides. The one is like direct development as antiviral drugs. And the second thing is to treat the symptomatic treatment like the respiratory distress and which is caused by the inflammatory response. So somehow we can suppress the inflammatory response that is produced due to the viral attack. So now this was the whole story. Then it says like what is COVID-19? COVID-19 is 89.1% similar. The genome of COVID-19 is 89.1% similar to SARS-like coronaviruses, and it has large RNA genome, and it exhibits unique replication strategy in which it utilizes non-segmental positive sense RNA genome and containing 5-cap structure. I mean, so basically all this thing causes the replicase polyproteins which are present in the uh, virus, which help in the translation of the genome of the virus. Its genome has already been sequenced. So there are 2/3 of the genome encodes for non-structural proteins. I mean, they don't make any structures. They are called NSPs. While 1/3 of the codes for 1/3 of the genome codes for structural and accessory proteins. What are those structural proteins? These are like spike protein, membrane protein, envelope, and nucleocapsid protein, SMEN. And then there are two viral replicase proteins. Again, the same thing that I mentioned earlier, like replicase polyproteins. There are two types of polyproteins, which are polyprotein A and polyprotein lab, which codes for 16 non-structural proteins. So this is the whole story of the virus. So we have like the structural proteins, which is which are produced by the 1/3 of the genome, while 2/3 of the genome produces the non-structural proteins, and non-structural proteins are actually produced in response to replicase proteins. And what is the mechanism of COVID-19? This is very beautiful. But first, virus binds to the host cell. Uh, like most commonly here, like the epithelial cells of the lungs. And then on the epithelial cells of the lungs, there is an enzyme which we know, which is very common, which is angiotensin converting enzyme 2, which is also a target for the antihypertensive drugs. So angiotensin converting enzyme 2 helps in the, so spike protein of the virus binds with ACE2. And once it binds with ACE2, then virus gets entry into the cell. So maybe I can show you in this diagram here what it is saying. Yeah, this. So this is basically angiotensin converting enzyme 2, and this is spike protein. So the spike protein binds to the angiotensin converting enzyme 2, and by the interaction of these three things like spike protein, angiotensin converting enzyme, this membrane is ruptured, and once the membrane is ruptured, the viral genome enters into the cell. Okay, so this is a target for us, spike protein. Well, I will discuss it later. And then once the genome enters into the cell, so it makes like it finds ribosomes of the cell and use those ribosomes to start making the proteins, right? So the translation happens, and then protein starts making. Okay, and then after that, a complete replication transcription complex is formed. That replication transcription complex makes other proteins, and then those proteins are again sent to the Golgi apparatus and the endoplasmic reticulum, and from there the Golgi vesicles are budded off in the form of viron, and then viron gets released. So once the new viron gets released into the cells, here comes the chemokines and cytokines. So a lot of chemokines and cytokines are produced when a new viron is released out of the cell. And that chemokines and cytokines start a cascade of inflammatory response that starts damaging the lung tissues. Once the lung tissue starts damaging, and then this is kind of a vicious cycle, and that ultimately results in the respiratory distress syndrome. So let me explain again. So after receptor recognition, viral genome is released into the cytoplasm production, and then once it happens, then there are like polyprotein A and polyprotein lab controls host ribosomes, and it starts finding the it starts forming the viral proteins. Then these polyproteins further form 16 non-structural proteins, and each has specific function. Briefly, they are important. They have important role in replication and transcription. And then M, E, and S proteins enter into the endoplasmic reticulum Golgi intermediate compartment and facilitate in the production of new viron. New viron elicits immune response, resulting in flooding of cytokines and chemokines, which destroy lung tissues, ultimately causing lung failure in severe cases. So this is the whole story of the COVID-19. Then is what is the target? So target is the virus itself. We can inhibit the virus itself by inhibiting its spike protein. If we destroy the spike protein of the virus, or we try to interfere in the replicative proteins like PPA and PP lab, we can destroy the virus. The second thing is uh, we make such type of siRNA or antisense oligonucleotides that can directly target the large genome of the virus. And finally, these two are the direct killing approaches. But the third approach is to mitigate the inflammatory response. So the inflammatory proteins that are being produced, somehow we can silence those proteins. And if we are able to silence those proteins, we can reduce the inflammatory response of the cells. Now, so these are the targets. Now, what are the challenges? The challenges, so he explained the challenges by giving example of the HIV. So basically they developed a system for the HIV in which they tried three approaches. First, they made the siRNA to target HIV regulatory proteins, which are TAT and REV. And then they also make like nuclear localizing RNA decoy to sequester the TAT protein into the nucleolus. And then they also used the ribozyme targeting CCR5, a chemokine receptor on WBCs. So this is the same thing, like if we target this CCR5, or we have to figure out like which chemokines are responsible in mediating the inflammatory response, so we can also reduce the inflammatory response of the virus. Now, lastly, after giving this all overview, it explains about what are the hypothetical approaches available to us using the oligonucleotides, which can be used for the therapy of the coronaviruses, or basically the COVID-19. I hope it is clear up till now. It's very simple. Eva.
Yes, it's pretty clear.
Yeah, and JJ? JJ.
Sorry, I was muted. Can you hear me?
Oh yeah, no problem. Yeah.
Yeah.
So, what do you understand? Just my simple question. What do you understand by silencing?
So basically, you want to prevent the part of the gene from, becoming a protein.
Yeah, that's good. That's correct. So basically, what happens? We know very simple process that the DNA is transcribed to make messenger RNA, and then messenger RNA is translated to make protein. So the target for the silencing is messenger RNA. So basically, siRNA destroys the messenger RNA using the silent risk, which is uh, RNA-induced silencing complex. These are the 5 different proteins that actually binds to the messenger RNA, and siRNA and messenger RNA binds in the presence of these 5 proteins and destroy the messenger RNA. And once the messenger RNA is destroyed, the protein is not formed. For instance, if we are talking about the spike protein, so what we have to see, first of all, spike protein is a very large protein. So we have to identify some regions of that. We have to go back to its genome or its gene, and we have to see in the gene which region is important for its activity. Then we design siRNA based on that region, and the cell starts making the messenger RNA. Then we give the siRNA. siRNA binds complementary to the messenger RNA and destroy the messenger RNA, and then the protein is lost. The same approach everywhere. So if we want to destroy the polyproteins A or polyprotein lab, so we'll have to figure out, like, we have to design the siRNAs. So the previously, like the first paper that I sent, in which they mentioned about 35 patents. So those 35 patents are basically about this siRNA thing, like which type of siRNA they used, what was their sequences, and which target proteins they used for the treatment of the COVID-19 or other coronaviruses. So if you understand the basic, then the hypothetical approaches will become, and then there is like aptamers. So you should know what are the aptamers. Aptamers are similar to siRNA, but they are longer than siRNA. So they usually the siRNA has 21 to 24 base pairs, while the aptamers have like 100 nucleotides, and they are specifically produced with a technique which is called CLEX, and they have very high affinity for their target compounds. The purpose is the same. They bind to the specific sequence, and then they make it ineffective. Like, let's make that sequence ineffective. Okay, let's see what are the hypothetical approaches. The first is like highly conserved regions within the replicase polyproteins. Encoding regions of SARS-CoV genome could make particularly attractive target for the gene silencing. So we know that this coronavirus is the variant of the previous coronaviruses, but we also saw that 89.1% of the genome is similar. So it means that there are some regions which has not been changed, and they are similar in all the coronaviruses. So those regions are called highly conserved regions. So we have to find those regions in the replicase polyprotein encoding regions, and then we can try to silence those. We can try to make messenger siRNAs and try to silence those portions. Second thing is that we can silence the angiotensin converting enzyme 2, because this is also a receptor for the spike protein. In infection susceptible tissues, this can be another target. Third target is the targeting of the most, and then there are problematic proteins which are like are its structural proteins like MES, which are generally responsible for patient immune response. In some cases, so strong that patient die from lung inflammation and CD8 depletion. We can also target the structural proteins, MES. Then we can also use aptamers, again, angiotensin converting enzyme 2, This prospect is bolstered by relatively fast development time available to aptamers through protein-based selects, which can reveal sequence candidates with high target specificity and sensitivity in relatively short amount of time. Then is aptamers conjugated to siRNAs or microRNAs can be used. So they are like now they are trying the dual approach in which they are using the aptamers as well as siRNA. The aptamers conjugated to siRNA or aptamers conjugated to microRNAs can be used to selectively target these antiviral agents. Target these antiviral proteins to target cells and tissues, offering an avenue of delivery for silencing strategies previously outlined. Same thing. Then again, this is a combination approach that we can use aptamers with antiviral analogs for the treatment of for the powerful approach to therapeutic treatment and should be considered for coronavirus therapies. Microorganisms with potential to down-regulate viral proteins and host gene responses that support infection can be identified via RNA sequence analysis followed by informatics. Once simulated, a therapeutic strategy can be devised wherein these microRNAs can be ectopically delivered to coronavirus infected cells. So this is approach, but what are the challenges? The first challenge is route of administration. We know that the problem with the gene therapy is that, so once we administer the genes in the systemic circulation, they are rapidly destroyed by the nucleases present in the blood or in the plasma. So this is a major target. This is a major problem. So as of now, with all the gene therapies, so we from that we can say that they are not stable. So if we somehow use some other routes, for instance, we are targeting the lungs, so we can start making the inhalational. We can exploit the inhalational route, right? Or we can use aerosol formulation or something like that. This is one thing. Then another thing is that we have to improve the serum stability and improve the pharmacokinetic profile. The third and most important thing I didn't mention maybe, and it is very common, and which is the entry into cells, for which we use polymers, peptides, CPPs. So this is the whole story. Is it clear or any confusion? So they just like this information they published, and it is published in Tier 1 journal, which is very esteemed journal. So any thoughts about this information? Dr. Tiwari, you have any question?
Not questions. So basically, it is more about sharing, about virus. What I understand that this paper share about virus and the target oligos can be used for that. So so that sounds okay. I think, and JJ already did some more reading and he presented last week to me. I think I did not record it, or I might have recorded his presentation. So he already go through all the different classes of medications and how they work.
Just interrupt you. So there are like the different targets, like
Yeah.
this diagram explain like
Yes.
where the chloroquine, hydroxychloroquine works, tapinavir, ritonavir, and then this is the most like this is the one which is like very buzz drug, remdesivir. So it actually does what it blocks the assembly of the virus.
Yeah, that's true. So now if you want to get started with writing, so I will suggest you two things. First, make a outline and divide the work to Eva and JJ. They both are the best student from our school, and so you have good chance. JJ is another few more weeks with us, so he's like you can ask him. He's crazy in working, so you just need to give him some clear instructions what you are looking for, and make sure he understand what you are looking for. Then he will write it very quickly. And with the Eva, same thing. So make an outline and divide the work. They both knows, I think, JJ, I don't know whether JJ knows how to use EndNote, because EndNote is very important. Otherwise, just give him a tutorial, or JJ, just join me. I am writing a paper, so I'm adding a EndNote to EndNote means you are writing citation using EndNote. So I'm doing it now. I can show you, even both of you, how to do that. So it's a pretty simple, easy software and web-based program that Chapman has it. But that helps you to write many citations, because every research journal has their own style. They want to write first name, initial, journal name, italic, bold, period. They're too complicated. So I can show you. So that's my idea. I like the idea, but this work need to be done as soon as possible, because if you are making it, somebody publish it, then you will be the second line. So then we need to change journal and strategy. So get started.
So we have the Zoom session as well. Like I think I gave to Eva. I can send to JJ as well regarding the EndNote if he is
Sure, sure,
interested.
sure. And I'm doing right now. Like,
Yeah, yeah. Then that's you can
they
have
want
like
to see, I
live
can show you in
live.
five minutes. So I can show that. So Imran, I suggest you to make a outline.
No,
Okay.
I have already the outline. I shared. I think I have shared already.
No, this
Uh,
is what you are reading from that paper, right?
no, no. In the previous email actually
Oh,
I also sent the outline. So this is
okay.
the outline.
Mm-hmm.
Yeah. So this is the outline. I think Eva and JJ should have this outline.
Yeah, I saw this already.
Yeah.
Okay, okay,
So
okay.
basically, this is my outline,
Mm-hmm.
siRNA therapeutics for the therapy of COVID-19 and other coronaviruses.
Mm-hmm.
So this is basically the title.
Mm-hmm.
And then we have introduction,
Mm-hmm.
then siRNA therapeutics, then siRNA therapeutics for the therapy of viruses. So it covers almost all viruses.
That's too much. It doesn't.
It's not, so let me finish and then I explain. So then siRNA therapeutics and coronaviruses. Then we have to review the patents approved for the treatment of the coronaviruses and COVID-19. And then challenges in siRNA-based viral therapies, which include stability, few animal models, comparatively long duration in translation from lab to bedside, how to address those challenges. And then ongoing work being done in this field, innovation and future prospects, references. This is the outline. So what I am saying is that, like, let me explain how we are going to do it. For instance, I explained that what I will write in the introduction. So this introduction will focus on siRNA overview. So we will explain what is siRNA, how it came into, particularly it answers what is siRNA, when was introduced, its potential for therapy of viruses, problem statement that COVID-19 and other coronaviruses, health and economic burden. And this point should be strengthened by the statistics. We have to look for the statistics of other viruses and coronaviruses and COVID-19, how much health and economic burden they have caused. Then few treatment options available for other coronaviruses, uh, but there is no approved treatment for the COVID-19, and no review published in this area so far. However, it is promising field for the therapy of COVID-19. The scope is to present the work done so far in this field. What are challenges, bottlenecks, how to address these challenges, a review of patents approved, future prospects and innovation. So this is the paragraph. I mean, I can write it. Then when I say siRNA therapeutics for the therapy of viruses, we don't have to explain it too much. We just have to, like, for instance, herpes simplex virus, HIV, we have to make a table in which, for instance, I can show you. So we just have to make a table in which we just uh, explain the virus, their target protein, as well as the siRNA that you used.
I
Okay.
think I know what you are talking about. You sent out another paper. They kind of did that too.
Yeah.
Okay.
Yeah. So this is basically, that's what I'm trying to dig out. If you have it, that's wonderful. Yeah. So basically, I'm just trying to focus on different viruses. The purpose is just to explain that siRNA is very much effective and it has been used for the treatment of the viruses. So aptamers. If you see in another paper, I have it. I don't know I shared it or not, in which there are like I think 6 or 7 drugs which are aptamer based, and they are approved by FDA.
Mm.
So I just want to emphasize the importance of the siRNA. So what I will write in sentences is that the importance of the siRNA therapeutics in viral therapy, and then I will give a table, and I will mention in the text that this table represents the major therapeutics, siRNA based therapeutics for the treatment of viruses. Then we will shift in the next thing. We will just focus on the coronaviruses.
Imran, one very quick note.
Mm-hmm.
Do you have the name of the drugs? Those are in the market from the siRNA based drug that
So siRNA is like one drug, which is
one petro.
yeah. But for aptamers, there are
So my suggestion is that you need to have some evidence to start your story. To
Mhm,
make a table, that's fine, but focus or explain maybe aptamer or some drug that
mhm,
is from siRNA to make a story more interesting, that this is a way it's going to come out.
mhm.
Okay.
Yeah.
Yeah, go ahead on your outline. Yeah.
Okay. So then we will focus on the coronaviruses particularly.
Mm-hmm.
And in this section, we will explain different coronaviruses. So here we have to explain like what are coronaviruses, and here we can also design a timeline, and then we go to the COVID-19. So we have to think creatively to make a timeline. After classification of the viruses, we will focus on the siRNA sequences with their targets and results. This will be presented as table. And then we have 35 patents. This is very simple. This section is very simple. We have 35 patents. We just have to read those patents and get the information out of that. I'm sorry. Let me see what I am going to show you a table in which it mentions the uh, uh, yeah, this one. No, this is a patent review. So this is just a paper which reviews only the patents. But this is anti-SARS coronaviruses. But we can, like, this is the model for us that how we review the patents. But I am looking for, I think I sent the email. In that email, we have that table. Uh,
It's okay. You can look
yeah,
at.
so there are like 35 patents. There are 35 patents.
So my suggestion is that if you have to get a summary or highlight of all 35, then divides some to Eva, some to JJ,
Yeah.
and give them, read one patent and know, because in patent there too much information. There is a lot of story background given, but where is the most important part? What is the result? What is the sequence? What is a oligo,
Yeah.
right? What was the target? So those things they need to highlight, and maybe write like 5 sentences, 7 sentences, not like 2 pages or 3 pages.
Yeah.
Okay, so
So
show them and how they can find the full patent. Okay,
yeah,
so show them, give
I
them
will show. Yeah, I
and
will show
focus
them.
with them. Yeah.
So maybe we can have a session tomorrow regarding the patents, like how to read a patent and then how to get the information from the patent. And then we will go to the next point, which is challenges in the siRNA-based viral therapies, which includes like stability. It's a big problem, stability in the serum. And the half-life is very small. It's like what I saw is like around 2 hours. And then there are fewer animal models. And then mm, from translation, like from lab to bedside, there is long time. Although the paper says like it's easy, it's very easy to design the siRNA based therapeutic and to perform the cellular studies. It's very easy. The problem comes from the cellular work to the translation into the clinical setting. It takes a long time. So somehow we have to see how can we reduce this time from the cellular work. So we perform the experiment in the cells, and cells say, okay, this protein has been silenced. The data says. But once we administer in vivo, then there comes the pharmacokinetics problems and the route of administration problems. And because of that, uh, we have seen like very few therapies in the market. and how can we address these challenges? Here we need extensive reading. So I have some data, I have some papers. So I mean, I can write this myself, and I mean, I can write 5, 6, 7. Okay. And, but I want the help from Eva and JJ regarding this. Like, if, yeah, we need a table here in section 2. Then we need a table here in section 3. And then we need to know what are different coronaviruses, right? Also, I forgot to mention here, like the role of carriers in gene therapy, where we have to, for instance, when I say carrier, like CPPs, polymers, we can also make some table about it. Polymers that have been used for delivering gene-based or oligo-based, gene-based or oligo-based, gene-based, oligo-based, the same term, based therapies into cells. We can play with it, yeah.
Okay, sounds good. I think you need to write where who has to work. Okay.
So let me open the to Eva and JJ, and they have studied it already. So what are their thoughts about which section they can do and what comes in their mind regarding this outline?
I like the outline. I like Dr. Tiwari's idea of not starting so grand. Start with like specific ideas and expand on what we can potentially use.
Mm-hmm.
And second thing, I think I can help with reviewing the patents and making the table. I think I can do that part, and you can just let me know how to read a patent, and I will extract that information and make it to a chart and paragraphs.
Wonderful. So I will do it tomorrow. Yeah.
Okay.
We will do it tomorrow at around the same time, right?
And regarding the table you mentioned for the first part, I'm not sure if you still want to do all viruses or just targeting like the related ones.
There are many reviews. We have data. This is not difficult. It appears difficult, but it's not difficult, right? So because there are, I can send you the papers, and I can make it very specific that this is how we have to make it.
Okay.
Yeah.
So I think I can try to do those 2 parts.
Okay, 2 and 4, right?
Mm-hmm.
And JJ, what is your
thoughts? Yeah, I would say same thing. I could help with those 2 and 4. But my question is, are we addressing just siRNA? Or are we like also saying aptamers included in that?
Yeah, I think we have to figure out this because they are closely related to each other, and our more emphasis will be on the siRNA, but we can also touch the aptamers as well. So
and then I had a question about number 3. So when you mean timeline, I'm kind of unclear, like what kind of timeline you're.
Yeah, what I'm saying is this. So basically, we need to know what was the first coronavirus. It is discovered by some lady in probably 1980s.
Yeah. So I think
just like a history of all the coronaviruses.
Yeah.
Early 2000 or when they started looking. So I think JJ, you can look into number 3, siRNA therapeutics. Yeah. So basically siRNA therapeutics and coronavirus, I think they are siRNA therapeutics are very common, but what are used, I think they are in a patent. There is nothing like in publication you may find, maybe very hard to find. So I agree with you. I think, Imran, you need to be very more specific, more clear what you are looking from this more number 3, what exactly you are looking for, because you may have a lot of information from other papers. Yeah.
First, I want to make a creatively timeline. Okay. I will show you the model of the timeline that I have in my mind. And then the 2 paragraph or 1 paragraph regarding the classification of the coronaviruses, like how from where it comes and all that stuff, right?
Yeah, it is.
And then we have to see again the siRNA sequences, their targets and results, and then table. So basically I'm looking for a timeline, classification, and a table.
So when these patents, as I think the patents are for basically mostly the patents are there, and patents usually have siRNA that target particular.
Yeah. So
You know.
basically, it
So
comes
what's
from
the difference between 3 and 4 if 4 are the patents with the sequence, which are siRNA?
basically, so there are also, aside from patents, there are some other as well, the siRNA sequences and their targets, and there are some publications as well. Providing you the papers is my responsibility. I will provide you the papers. Reading those papers and getting the information and tabulating this is your responsibility.
Mm.
Okay. Okay. Yeah. So just to give them information what they are looking to know what is the target protein or gene they are silencing.
Yeah.
What was the sequence? How long was oligos, or what specific delivery system they use?
Yeah.
What kind of animal model they used in their research, or they use the cell-based model? What was their endpoints of conclusion? So okay.
Eva, does it make sense now?
Mhm.
Yeah.
Yeah, I think it will make more sense if you can show us like a patent and.
No, I mean I changed the section 3 to 4. Like I changed the section 3 and 4.
Yeah, I think with this outline, you start looking into it, and as you proceed, maybe 1 or 2 days more, you may able to find some more information and see how to make it more better organized timeline. Because once you have a little bit more clear timeline, I can email some editor to make sure I say them we are working on it and we need their yes to submit this to their journal. Instead of writing everything, then approach them after 2 weeks, or I think it will be hard to finish it in 1 week, but let's see. I'm thinking about 2 weeks at least.
Yeah.
So, or 3 weeks maybe, I'm not sure. So once we finish up and then try to say them, there may be already a couple of people may couple of journal may show up, there is new article coming up. So I think once we have a very clear outline, I have to reach out to the editors to say that we are interested in submitting this review to their journal and we need their approval, so they understand we are in a queue basically.
So for now, let's see. Anti-SARS coronavirus agent patent review. I just want, JJ, or if Eva is interested. So I just want here. I mean, you see this.
So my suggestion is that if you want to continue, you continue. I have to catch up other meeting.
Okay.
So you can continue, and once you finish up and JJ and Eva want to do some end note, I am doing it now, so I can show them. Once you finish up, so just let me know. Okay. So I will be quiet now, and then you guys will continue.
Okay.
Take care.
Okay, thank you. So, for instance, this is a review of all the patents regarding the anti-SARS agents, and it has many things like, so we are not interested in different things. We are just interested in these RNA products. So if you see this RNA product, so we have reference number 60. I will go directly to the reference number 60. I will check what is written there and how this author got the information from reference 60 and wrote it here. So, I mean, by this way, you will understand, oh, this was written and he wrote it this way. Okay. So basically, you see functional receptor for SARS coronavirus nature 2003, right? So we have to go there and then we have to see how he wrote it. And here you will enjoy. I mean, I actually enjoyed. So basically, you will see that how authors work. Right. So basically going for this line, I am more interested in reference number 60 rather than this line. Similarly, this is all about angiotensin converting enzyme 2. Then this, again, a Chinese patent now 64. So we have to go to 64 and see what is written there. So I will explain how the challenge. And for instance, here it says, this is a beautiful line. The designed double-stranded RNA named siRNA-MIE has sequence of this, complementary to the sequence of M protein in mRNA 220 to 421 nucleotide. And then another siRNA-M2 which has this sequence, and it is complementary to 460 to 480 nucleotide of the M protein. These two siRNAs were shown to inhibit the expression level of M protein mRNA. But I want to know how much silencing was happened, how much inhibition happened, which cell lines did they use, and how did they come up with this result. And we can easily find that if we go and explore 64.
Mm-hmm.
You got it? So I mean, you can read this paper. I'm going, after the session, I'm going to send you some papers to read.
Mm-hmm.
So just read them. And then if you want, we can have a meeting in the evening today, or we can have meeting tomorrow.
Okay.
Yeah. JJ, do you have any like, what is your input about this review, or what, how much are you interested, and like, what are your thoughts?
Yeah, I think that would be good.
So we are actually addressing an issue which has affected all the world.
Yeah.
Yeah. And I mean, if we spend some good time and creatively analyze this thing, this can be a very, very good thing.
Yeah.
Okay, I have not seen you, JJ. Can you introduce yourself?
Yeah. So I'm a P3, so last year of a pharmacy program.
Mm-hmm.
Let me see if I can.
Yeah.
Here. In the office.
मैं भी आ रहा हूँ. Excuse, I am busy. Yeah, sorry.
So, yeah, I'm on my last few weeks of being at Chapman. So I'm almost done.
Okay, that's great. That's great. So okay, so let's see. I'm going to send you email right after this session, and I will share with you some papers. And meanwhile, you can also dig some, like you can Google, like maybe you can use PubMed, Science Direct, or scholar.google.com. That what are the papers that we come across regarding the siRNA or oligonucleotide based coronaviruses, and like try to explore yourself some papers. You know how to explore the papers.
Yeah.
Okay. So Eva also know that. So today, just read the papers that I'm sending, and then let's see what happens tomorrow.
Okay.
Yeah. All right.
Mm-hmm.
Thank you.
Sounds good.
All right. See you then.
Yep.
Okay.
Okay. Thank you.
Thank you.
Thank you.
Bye.